To all the students. So yes, today we are in our last session of our biological classification. So far we have dealt with 5 kingdom system of classification. Kingdom Monera is done, Protista is done, fungi up till classification level it is done. Today we will look into each class of fungi in detail. After that, kingdom Plantae and Animalia in brief account, wherein we learn it in more detailed way under the chapters plant kingdom and animal kingdom. And after that, we are going to look into those organisms which are not kept under 5 kingdom system of classification. Clear, right? Yeah. Let's get started. First is what? Phycomycetes. Phycomycetes is the primitive form of fungi. Sit. Phycomycetes is the primitive form of fungi. This is also called as algal fungi. Fine. These are found in aquatic habitats on decaying wood and as what? Obligate parasites. Okay. Mycelium is what? Aseptate mycelium we have learnt yesterday. Aseptate mycelium, can I say it's xenocytic? Xenocytic, yes or no? Yeah. Aseptate xenocytic condition. Aseptate xenocytic condition. Okay, where multinucleated condition is observed, and asexual reproduction is through zoospores and aplanospores in Phycomycetes. Remember, asexual spores and sexual spores, they are very important. Every alternate year, if you are getting a question from fungi, most probably the question will be from asexual spores and sexual spores. It should be registered in your mind. Okay, asexual spores of Phycomycetes are what? Zoospores and aplanospores. Amongst these two, which is motile and which is non-motile? Zoospore is motile, aplanospores is non-motile. Okay, and then here you see sexual reproduction. Sexual reproduction is of course through fusion of two gametes, but in Phycomycetes, advanced mode of sexual reproduction will not happen. Advanced mode of sexual reproduction includes what? Plasmogamy, karyogamy and meiosis. These 3 steps will happen only in the Ascomycetes and Basidiomycetes. But the sexual reproduction in Phycomycetes, this happens in 3 ways, any of the 3 modes, either through isogamy, anisogamy or oogamy. That is fusion of gametes. Fusion of gametes in isogamy means what, ma? 2 gametes with the same size, structural similarity will be there and physiological similarity also will be there. Anisogamy, they can differ either in structure or in the physiology. And oogamy, one small motile male gamete gets fused with large non-motile female gamete. So, sexual reproduction in Phycomycetes can be in any of these 3 modes. After the fusion of 2 gametes, you tell me what is the ploidy of the fungal hyphae? What is the ploidy of fungal hyphae? Yesterday we have discussed. What is the ploidy of fungal hyphae? Yes, haplontic body. Fungal body is a haplontic body. It has haploid hyphae. So, gametes are what? Both female and male gametes are haploid. Okay. Post-fusion, the products which are formed in the oomycetes are oospore, zygomycetes is zygospore. Okay. These are what? Diploid, and this is the region where meiosis happens. Meiosis is what type we have discussed? Zygotic meiosis. When you are looking into the plant kingdom, algae is having what again? Algal division is having zygotic meiosis. In algae, zygotic meiosis only will take place, where in Bryo, Pterido, Gymnoangio, sporic meiosis will occur. Same as fungi is also thalloid plant body. Okay, algae is also, fungi is also thalloid body, algae is also thalloid plant body. So, this thallophyta members, in the thallophyta members, zygotic meiosis will occur. Is that clear? Yes. So, here zygotic meiosis occurs, and after the meiosis, the spores which are formed are haploid, which will develop into a new hyphae. So, what all you have to remember from this slide beta? Asexual reproduction, previously asexual reproduction in Phycomycetes is through zoospores and aplanospores, and this is the primitive form of fungi where you see aseptate condition, that is xenocytic or multinucleated condition. This is also called as algal fungi. Okay. Asexual reproduction is through zoospores and aplanospores. Sexual reproduction is through any of these 3 modes, that is isogamy, anisogamy and oogamy. After that, the products which are formed in oomycetes and zygomycetes, these are what are subclasses under Phycomycetes. Okay, in Oomycetes the spore is oospore, Zygomycetes it is zygospore. This is the site of meiosis wherein haploid spores are formed, and this haploid spores will develop into a new hyphae. This new hyphae is haploid. Okay, clear, understood? Next. Yeah. So, what are the examples of Phycomycetes? To remember the examples of Phycomycetes, there is a mnemonic which I gave you, RAM, RAM, wherein R is Rhizopus, commonly called as bread mold. Okay, Rhizopus, which is called as bread mold. So, RAM, Rhizopus, which is called as bread mold. A is what? Albugo, which causes white spots on the mustard leaves, and M is mucor. Okay, clear, right? Rhizopus, albugo and mucor, of which Rhizopus and mucor belongs to Zygomycetes. Albugo belongs to Oomycetes. Oomycetes includes what, ma? Albugo. Clear, right? Clear with Phycomycetes, ma? Any doubt anywhere? No, right? Yeah. Next is what? Ascomycetes. Ascomycetes in Ascomycetes you can observe varied amount of morphological structures. It can vary from unicellular yeast to multicellular structures like penicillium, claviceps and all. So, example for unicellular form is what? Unicellular yeast, Saccharomyces cerevisiae, and multicellular forms include example penicillium. Few more examples: claviceps, aspergillus, okay, aspergillus, okay, all these are different fungi which are multicellular under the class Ascomycetes. So, this Ascomycetes is commonly called as sac fungi because it has sac like ascocarps, okay, and the habitat, if you see, they are saprophytic, decomposers, parasitic and coprophilous. Coprophilous means those which live on dung, okay, and mycelium is septate. What type of septa you observe, beta? Pore like septa, we have discussed yesterday, right? Pore like septa is present. Fine. Everything is clear from here, no? Any doubt anywhere? Yeah. Next, asexual reproduction in Ascomycetes. So, in Ascomycetes, the asexual spores which are present are what? Conidia. We have discussed yesterday only that asexual spores, conidia. Only exogenously released asexual spore is what? Exogenously released asexual spore, conidia. We have discussed yesterday, right? So, here conidia from conidiophores will be dispersed out exogenously, okay. Special hyphae, those are what? From conidiophores, they produce conidia exogenously. Conidia on germination, conidia when they fall onto the suitable substratum, on germination they will develop into a new hyphae again. Understood, ma? Okay. So, here conidiophore is haploid, conidia are haploid, and that gives rise to a haploid mycelium. Okay, because only mitosis occurs here, there is no chance of meiosis. So, this is how they are exogenously released, ma. From the hyphae, the fruiting body conidiophore is there, you can see it, no. So, this conidiophore will have some structures called as sterigmata. Over the conidiophore fruiting body, what is present? Sterigmata. This sterigmata will release the conidia exogenously. Remember this structure, important, okay. What, with the help of what structures over the fruiting bodies conidia are released exogenously? If that is your question, the answer is what? Sterigmata. Clear, right? Yeah. Next, sexual reproduction. Sexual reproduction occurs through what? Yeah, sexual reproduction is from again specialized fruiting bodies, those are what? Ascocarps. From fungal hyphae, what happens initially? Plasmogamy occurs, two different strains, like we can consider positive strain and negative strain hyphae, they will fuse and first plasmogamy takes place. Second is karyogamy, the two nuclei will fuse. Okay. After karyogamy, the diploid nucleus which is present undergoes meiosis. Okay. This process happens inside what? Ascocarp. Okay, inside ascocarp what happens? Ascocarp, which is haploid, inside that antheridium and ascogonium will fuse. Okay, and here female fruiting bodies, the ascocarps, can be of many different structures. Ascocarp can be of cup or saucer shape, flask shape, globose shape. If it is cup or saucer shape, you will call it as apothecium. Ascocarp structure, it is called as what? Apothecium. If it is flask shaped, it is called as perithecium. If it is globose shape, it is called as clistothecium. Understood? Apothecium, perithecium and clistothecium. Clear? Okay. So here you see ascocarp, antheridium fuses with ascogonium and this forms ascus. Wherever you remember, like oospore and zygospore there in phycomycetes, I said you those are the sites of meiosis. Site of meiosis, it means it should be diploid. Okay. Here what is diploid in ascomycetes, Kunal? What is diploid? Ascus is diploid. Be here. Okay. Ascus is diploid. The plural form is asci. Okay. This is diploid and here zygotic meiosis will take place. After meiosis, spores will be released. These spores, which are haploid, landing onto the suitable substratum, they will develop into a new hyphae, haploid hyphae. Any doubt anywhere till here? Easy, no? Understood? Okay. You have to remember the different types or shapes of the ascocarps here. Fine? Next. Examples. To remember the examples of ascomycetes, there is a mnemonic which I can give you, that is ACP-NT. Okay, ACP-NT. Told you many n number of times. A is for Aspergillus, C is Claviceps, P Penicillium, N Neurospora, M and T morels and truffles, and Y is for yeast. Saccharomyces cerevisiae. Okay, ACP and T, remember, for Aspergillus. Okay, clear with the class ascomycetes, ma? Any doubt anywhere? Yeah. Next is what? Basidiomycetes. Basidiomycetes class is called as advanced group of fungi or advanced class of fungi. Advanced class of fungi, because there are only sexual stages involved in the fungal members belonging to Basidiomycetes. There are nowhere asexual stages, you can see, nowhere asexual stages are present over here. They are commonly called as what? Club fungi, puff balls or bracket fungi. Okay, most common forms are what? Mushrooms, puff balls and bracket fungi. These are the most common forms. Okay, next. They grow in soils, on logs, tree stumps and in living bodies as parasites as well. Mycelium, if you see, mycelium is septate. What type of septa is present in Basidiomycetes, ma? Mycelium is septate and it has dolypore septa. Yesterday we have discussed, right? Dolypore septa. Okay. Ascomycetes is having what septa? Pore like septa. Basidiomycetes? Dolypore septa. Ascomycetes? Remember, be here. What happened to you people today? Okay, this is dolypore septa. Okay, dolypore septa is present. And next, reproduction, if you have to consider, vegetative mode of reproduction is of course fragmentation, binary fission, budding. Okay, those are the three modes of vegetative reproduction. Asexual reproduction is absent. Sexual reproduction is through basidia or basidiospores. Basidiospores or basidia. Okay, clear? Clear till here? Next. Sexual reproduction, here if you see, this is what? Basidiocarp. Okay, of course there are three stages of sexual reproduction. What are they, ma? First is plasmogamy. Good. Second is karyogamy.
Third is meiosis of spores.
Good. Karyogamy, and third is meiosis. What type of meiosis? Zygotic meiosis. So, plasmogamy is fusion of two different strains.
Plasmo
two different plasma or two different hyphae. This fusion happens inside the fruiting body, that is basidiocarp. Okay. Post fusion, what forms? Post fusion, if you see, yeah, post fusion after plasmogamy, you can see dikaryon structure. Dikaryon structure. Dikaryon is N plus N structure, and in karyogamy, fusion of this dikaryon will form what? Diploid cell. Okay. And what forms a diploid cell inside? Basidia, or basidium is singular, basidia is plural. Basidium or basidia, and here this basidium or basidia is the site of meiosis. Okay. And post meiosis, what happens, Nana? 4 spores will be released. Each spore is haploid, falling onto the suitable substrata that develops into a new individual. Okay. So here if you see, this is what? Basidiocarp, and this is basidium. Okay. Basidium is diploid. Remember, basidium or ascus is diploid, because those are the sites of what? Meiosis. They are formed after gametic fusion. Clear to everyone? Understood? Any doubt anywhere, ma? No, right? Yeah. Next. What is the site of meiosis in ascomycetes? Site of meiosis in basidiomycetes? Basidium or basidia. Okay. What are the sexual spores in the ascomycetes? Ascospores. Sexual spores in basidiomycetes? Basidiospores. Of amongst these two, which is exogenously released? Remember, again and again I am telling you, asexual spores, the only asexual spore which is exogenously released amongst the examples we have learned is conidia. Okay. The only sexual spore which is exogenously released is basidiospores. Asexual spore is conidia, sexual spore is basidiospores. Remembered? Now, fructification is large and conspicuous in basidiomycetes. Most advanced class of fungi, this is the most advanced class of fungi I told you, because it has fructification and which is large and conspicuous, and here only sexual stages are involved. Examples include agaricus, eustylago, puccinia, polyporus, lycoperdon. Okay. Eustylago is smut fungus, puccinia is the rust fungus. Okay. These are the examples under basidiomycetes. You can remember this, like a pulp will be the mnemonic for this. Okay. A pulp. Agaricus, puccinia, eustylago, lycoperdon, polyporus. Okay. This is the mnemonic for this. Deuteromycetes is imperfect fungi. Mycelium is septate or aseptate? Mycelium is septate or aseptate? Septate mycelium. Only aseptate you will see in the primitive class of the fungi. What is that? Phycomycetes. Only in Phycomycetes you will see aseptate condition. Deuteromycetes it is septate, and they are largely what? Decomposers. Few of them are saprophytic and parasitic. Okay. And they decomposers, they feed on what? Litter. Okay. And that act as dead and decaying matter. They will help in the mineral cycling accordingly because of their nutrition. Largely they are decomposers, they feed on the litter, dead and decaying matter, that will be helpful in what? Mineral recycling. Okay. Asexual reproduction takes place. Asexual reproduction is through conidia again, which is exogenously released spores. Clear, right? Conidia, sporangiospores, all types of spores will be there. Asexual reproduction, and there is no sexual reproduction. Okay. Here you see, while establishment, there is one paragraph in your NCERT, here is the detailed explanation of that. While establishment of linkages in between fungal classes, fungi with perfect sexual stages were moved on again to ascomycetes and basidiomycetes, which means like those fungi which are kept in deuteromycetes, again when they are cross checked, when they are observed the sexual stages present in those fungi, they are again moved back to what? Ascomycetes and basidiomycetes. That's it. You can expect some statement based questions from this area. Okay, and examples include Coletotrichum, Alternaria and Trichoderma, that is CAT, CAT, you can remember like that with that mnemonic, Coletotrichum, Alternaria and Trichoderma. So in this table I want to give you like a homework only, okay, all of you. I will anyhow send this PDF for you. Take this table as a homework, make this table in one sheet only, okay. So we will leave this fungal concept by once revising the examples, okay. What are the examples of phycomycetes? Examples of phycomycetes.
Rhizopus, albugo,
Now you tell me. Rhizopus, albugo, mucor.
Rhizopus and mucor are zygomycetes.
Yes, and albugo oomycetes. Ascomycetes examples you tell me. Only yeast. Remember the mnemonic first, right? Did you take down the mnemonic at least?
Aspergillus.
Are you taking down the mnemonics? ACP and
NTYM. Aspergillus.
Yeah, Aspergillus.
Claviceps. Penicillin.
Aspergillus, claviceps,
Penicillin. Neurospora.
Penicillin, Neurospora,
Truffles.
morels and truffles.
Truffles. Yeast morels.
Yeast. So amongst this, yeast is used both in brewing industries and baking industries. It is also called as brewer's yeast or baker's yeast. Morels and truffles are edible. Neurospora is used in the genetic works in the laboratories. Penicillin is the first antibiotic that is observed. Yeah, Penicillium notatum. And claviceps aspergillus. Okay, clear? Fine. Next, basidiomycetes. Mnemonic?
Apple.
Yeah, agaricus, polyporus, or you can take down Puccinia also here, your wish. Puccinia, Eustilago, Lycoperdon, and polyporus. Okay. Next, deuteromycetes, CAT. Okay. Coletotrichum, Alternaria, Trichoderma. Coletotrichum, Alternaria and Trichoderma. Clear? Okay. This table is a homework for you. Work it out. Okay. Any doubts you are having, you can ask me. Okay. Next. Kingdom Plantae in brief. Okay. Kingdom Plantae includes all the eukaryotic chlorophyll containing organisms. Cell wall of plants. Plant cell wall includes what ma? Cellulose, hemicellulose, cellulose, hemicellulose and pectin. Okay. Cell wall includes what? Cellulose, hemicellulose and pectin. Next, there are few important points with Kingdom Plantae is divided into 5 divisions. What are the 5 divisions of Kingdom Plantae? First is Thallophyta. Thallophyta includes?
Thallus-like body algae.
Thallophyta includes, usually Thallophyta includes algae and fungi. Thallophytic plant body, plant Thallophyta includes what is algae. So first division is algae. Second is Bryophyta.
Leaves not fully developed.
Yeah. Third is Pteridophyta.
Fourth is
Gymnosperms. Gymnosperms and Angiosperms. Gymnosperms and Angiosperms. Okay. In Plantae, in kingdom Plantae, you can observe alternation of generations. Okay, so you can see both haploid gametophytic phase altering with diploid sporophytic phase. Haploid gametophytic phase alters with diploid sporophytic phase. Okay. Nutrition is autotrophic, that we will discuss in the next slide. Few of them are parasitic also, I will tell you. This is clear, no? Okay. So, here you see nutrition, mostly it is 90% of the plant kingdom, plant members are autotrophic. They prepare their own food material. But then few members are partially heterotrophic, like insectivorous plants and parasites. Insectivorous plants includes bladderwort and Venus flytrap. Raghav, please move to this corner. Keep your bag that side. Insectivorous plants include what? Bladderwort and Venus flytrap. Parasitic plants include what? Cuscuta. So, in parasitic plants, you can see two types of plants beta. One is partial parasites and second is complete parasites. Okay. Partial parasites and second is complete parasites. Partial parasitic plants includes Viscum and Striga. Okay, which will dwell into only the xylem tissues of the preceding plants. They will absorb only water and minerals from the preceding plant. Whereas complete parasites will drive the nutrients, both food materials, okay, sugar molecules and water and minerals from the plant body, okay, which includes Cuscuta and Rafflesia. Cuscuta and Rafflesia. Clear? Understood? Reserve food material will be what? You have to tell me. Fine? Next. Kingdom Animalia. Animalia includes what? Heterotrophic, eukaryotic, multicellular organisms. There is no cell wall. What is the limiting boundary of animal cells? Plasma membrane. Nutrition is strictly heterotrophic, or you can also say holozoic mode of nutrition. Nutrition is heterotrophic or holozoic mode of nutrition. Okay. Reserve food material is glycogen. Reserve food material is glycogen. So, here you can see definite growth patterns and locomotion. Okay. You can observe elaborate sensory and neuromotor mechanism, which is not seen in the kingdom Plantae, and reproduction is sexual followed by embryological development. Okay. This is the key features of kingdom Plantae. Brief account, Animalia. You will learn it in more detail in animal kingdom. Okay. Next. Now, we will discuss about those organisms which are not included in the 5 kingdom system of classification by R.H. Whittaker. What are those? First is viruses, second is viroids, prions and lichens. First we will see viruses. These are not included in 5 kingdom system of classification. First let us observe about virus. Virus is most popularly known as infectious agents, or they can also known as living entities. They are also known as living entities. Okay. infectious agents or living entities. They are non-cellular organisms, okay. They are exceptions to cell theory, because cell theory is applicable to only those cells which has cell membrane and protoplasm, okay. Protoplasm includes nucleus and cytoplasm, single nucleus, cytoplasm and cell membrane. Only to those cells cell theory is applicable, but virus will just have a genetic material and a protein coat, genetic material and a protein coat. So, this is not acceptable, and virus will form connecting link between living and non-living entities, okay. These are infectious agents, living entities, non-cellular organisms. They are connecting link between living and non-living things. They are not applicable to cell theory as well. They are exceptions of cell theory. Next is virus outside the living organisms, they cannot replicate.
Cannot basic metabolism, cannot.
Yeah, there will not be any basic metabolism also. Inside the host or living cells, they can replicate and they can be metabolically active, okay. Clear, this is the basic description of the virus beta. And next, discoveries, most important segment, okay. First, virus is discovered as poisonous venom, okay. Virus is described as poisonous fluid or venom by Ivanovsky, okay, and he is the scientist who has extracted the fluid from the infected plant, okay, a plant which has tobacco mosaic disease, yeah, tobacco mosaic disease caused by what virus? Tobacco mosaic virus, okay. Tobacco mosaic virus is extracted in the form of a fluid from the infected leaves of tobacco mosaic plant, okay. This fluid is passed through bacterial filter paper. Fluid is passed through what? Bacterial filter paper, and he has observed that these particles are being moving from the bacterial filter paper. They are easily floated out from the bacterial filter paper. They can pass out through bacterial filter paper. This is the observation which is reported by whom? DJ Ivanovsky, okay. The infectious particles passes through, take down parallelly, bacterial filter paper. Fine, infectious particles passes through bacterial filter paper. Next it is W.M. Bajering. W.M. Bajering, who gave out the concept called as, contagium vivum fluidum. Contagium vivum fluidum, which means like the infectious particles or the infectious fluid from an infected plant, when it is transferred to a healthy plant, it has carried forward the infection from the infected plant to healthy plant, okay, understood. Infectious fluid has carried forward the disease to a healthy plant. Fine. And next it is W.M. Stanley. W.M. Stanley has crystallized the virus into small protein particles or crystal proteins, okay. Crystallization of virus into particles. Crystallization of virus particles. Fine. So, you have to remember these three discoveries. First is what, Nana? DJ Ivanovsky, where virus is a poisonous virus is noted or discovered like a poisonous fluid or venom, where the infectious fluid which is taken from the tobacco mosaic plant, when it is transferred through a bacterial filter paper, it has passed out through the bacterial filter paper. That is the observation of D.J. Ivanovsky. Second, it is W.M. Bajering. W.M. Bajering gave out the concept of contagium vivum fluidum, okay, wherein the infectious fluid causes the disease to the healthy plant, okay, and W.M. Stanley crystallized the virus into particles, okay, clear? Next, virus usually has a genetic material and a protein coat, you know up till here, but few of the viruses will have an envelope over this protein coat, the envelope which is made up of polysaccharides, lipids and proteins, okay. So, few of the viruses will have what ma? Envelope, capsid and genetic material, okay. Enveloped viruses we will call it as envelope, capsid and genetic material. Okay, envelope, capsid and genetic material. So, this envelope is made up of what? Polysaccharides, lipids, yeah, proteins. Few of the enveloped viruses examples include herpes virus, coronavirus, influenza virus, all these are the examples of what? Enveloped viruses. Now about capsid. Capsid is purely made up of proteinaceous coats. Proteins are made up of what? Amino acids. The protein structures which are present in the capsid or the protein subunits which are present in the capsid are called as capsomeres, okay. Capsid is made up of protein, okay. Protein subunits which are present in the capsid is called as what? Capsomeres. These capsomeres are arranged in helical, polyhedral or geometric forms. Capsomeres are arranged in what? Helical, polyhedral and geometric forms. Clear, no? Any doubt till here? Yeah. Next. Nucleoid can have either DNA or RNA. Based on the organism it is causing the infection, okay. So, animal infecting viruses includes what? Animal infecting viruses includes double stranded DNA, that is pox virus and herpes virus, okay. It includes double stranded DNA, that is pox virus and herpes virus. Plant infecting viruses includes what? Single stranded RNA. Examples include TMV, not HIV, it is TMV, tobacco mosaic virus. And then bacterial viruses or bacteriophages includes double stranded circular DNA. Clear with the nucleoid or xenophore or the genetic material? Genetic material is based on the organism it is causing the infection. Viruses which are infecting plants will have what genetic material? Single stranded RNA. Viruses which cause infection in the animals? Double stranded DNA. Viruses which cause the infection in bacteria? Double stranded circular DNA. Yes, Raghav? HIV. Yes, RNA virus, that is retrovirus, which has RNA as the genetic material. Next. Yeah, yes. Retroviruses which has RNA as the genetic material. Okay? Clear? Next.
But it is animal infecting, not plant.
See, the statements which are passed out are not unanimous. There are some exceptions. In molecular biology, we have learnt that in few organisms, RNA is the genetic material and they will synthesize DNA from RNA. Didn't we learn? That process is called as reverse transcription. By the enzyme reverse transcriptase. Okay, so HIV belongs to that example. We can add on that example to that. Okay, good. Next, viral infections. Okay, in animals, the infections which are caused includes mumps, chicken pox, herpes, influenza and AIDS. In plants, you can see tobacco mosaic disease which is caused by the virus tobacco mosaic virus, TMV. and cauliflower mosaic virus by cauliflower mosaic disease by cauliflower mosaic virus. It is as simple as it is. Okay. Next, symptoms of viral infections. Symptoms of viral infections includes mosaic formation, leaf rolling, leaf curling and yellowing, vein clearing, warping and stunted growth. These are the symptoms of viral infection. Clear? Now about tobacco mosaic virus. Tobacco mosaic virus, as we have discussed, it is first observed by whom? DJ Ivanovsky. And how to describe the structure of TMV, ma? This is an elongated rod like structure, okay, which has 3000 angstroms long and 180 angstroms in diameter. Molecular weight of the tobacco mosaic virus is 39.4 into 10 to the power of 6 daltons. And the capsid is made up of protein subunits. Capsid will have protein subunits. Those are what? Capsomeres. These capsomeres are helically arranged. Okay, helically arranged capsomeres. And then the genetic material is what? Single stranded RNA. RNA is made up of nucleotides. How many nucleotides over here? 6400 nucleotides. So if you have to take a ratio between the number of nucleotides and number of capsomeres present in the TMV, what will be the ratio? It is 3:1. 3:1 ratio. 3:1 is like 6400 nucleotides is to 2130 capsomeres. Understood, Kunal? Any doubt anywhere? Question mark? Huh? Clear with TMV, right? Yeah. Bacteriophage. Has what genetic material? Double-stranded DNA. You can describe it structurally as tadpole structure. Okay, it can be described as what? Tadpole structure. It has a polyhedral head and a helical tail. Polyhedral head and a helical tail. The head is having what? Double-stranded DNA, and it has a neck followed by tail, which is helically arranged, and at the end it has what? Protein enzymes. Tail fibers will have some enzymes like lysozyme. This lysozyme will degrade the wall layer of the bacteria whenever it is causing the infection. Clear with the structural description of bacteriophage? Any doubt anywhere? No, right, ma? Next. Subviral agents includes viroids, prions. Okay. Remember, prions means abnormally folded proteins. PP you can remember easy, no? Prions, abnormally folded proteins. Viroids are just having genetic material, that is single stranded RNA. Viroids have genetic material. So here if you see virus, viroids are nothing but the virus without protein coat. Okay. This is given by the scientist T.O. Dioneer in the year 1971. They caused the disease potato spindle tuber disease. It has what? As it is infecting the plants, it has single stranded RNA which is tightly folded to form circular or linear structures sometimes. They have low molecular weight when compared to virus. Okay, low molecular weight when compared to virus. So listen here, when the molecular weight is considered, ma, first it is virus. After that, next is viroids. In the descending order I am showing you. Lastly is prions. Because prions are abnormally folded proteins. Proteins are made up of amino acids. But genetic material which is present in viroids is made up of nucleotides. Each nucleotide is again composed of sugar, phosphate group and nitrogen base. Virus has both protein coat and the genetic material. The logic is as simple as it is, right? So according to the molecular weight, the first molecular weight or the highest molecular weight is for virus, then viroids, then prions. Have to remember this. Okay, this can be asked with examples also. Here he can give TMV, tobacco mosaic virus. Here he can give potato spindle tuber virus or viroid, and in prions he can give you CR-Jacob disease. You should observe the diseases and you have to note it, whether it is a virus, viroid or prion. Okay. Next. Prions you see, ma. Prions are what? Abnormally folded proteins. I already told you the memory hack also. Prions, proteins, you can remember like that. Nucleic acid is completely absent. The major infection or the target infection is on the nerves. So this causes most of the neurological disorders. In sheep though, we will call it as scrape disease, which is caused by prions, and mad cow disease in cattle, which is called as bovine spongiform encephalopathy. Okay, mad cow disease is called as what? Bovine spongiform encephalopathy. Okay, bovine spongiform encephalopathy, and this has an analogous variant, that is CR-Jacob disease in humans. Okay, the analogous disease of mad cow disease which occurs in humans is called as CR-Jacob disease. Clear? So these are the two variants. This is bovine prion protein, this is human prion protein. Can you see the variant, analogous variants? Next. Molecular weight in the ascending order. First is prions, then viroids and viruses. Okay. So you are clear with virus, viroids, prions. Last segment, that is lichens from biological classification. So lichens include symbiotic association between algae and fungi. They are very good pollution indicators. Okay. Lichens are good pollution indicators. How I will tell you. Now, no, I will tell you. So lichen will have two components, algal component and fungal component. Okay, algal component is just 5%, but it provides the nutrition for entire fungi which is there in the bulk amount. Okay, fungi will in turn provide the shelter to algae. Okay, next. Algal component is called as what? Algal component in lichen is called as what? Phycobiont. Fungal component is mycobiont. Okay. Next. Here you see they are good pollution indicators. They are good pollution indicators, especially for sulfur dioxide pollution. How you see the internal mechanism? This will not come in your NEET examination, but for your knowledge I am telling you. Okay. So lichens have algal component, algae will have chlorophyll. Chlorophyll, you know, it has what? It has a porphyrin ring and a phytol chain. Okay. This is a phytol chain and this porphyrin ring. Porphyrin ring and phytol chain. At the center of the porphyrin ring, it has what? Magnesium. Magnesium is connected. Okay. So whenever it is exposed to the pollution, be it nitrogen dioxides or sulfates, sulfur dioxides and all, the bonds between carbon and magnesium will be cleaved. This will lead to the structural disability of the chlorophyll. When the structure of the chlorophyll is dismantled, the physiology of chlorophyll will be dismantled. When algae is not providing the nutrition to fungi, there is no point of the lichen's existence. Understood everyone? So this is the internal physiology of it. That is how we can term the lichen as what? Good pollution indicators. This is more sensitive to the air pollution, especially sulfur dioxide pollution, and they don't grow in the polluted areas. This is the reason for that. Okay. So that's all for today's session, ma. Complete biological classification is done. So in our next class, what we will do is, I have set up 50 questions from biological classification. Advanced level questions, I have included diagram based, statement based questions, match the following questions and NEET PYQs as well. Altogether 50 questions we will exercise in our coming class, biological classification. After that we will move to the chapter plant kingdom. Is that clear? Okay. So based on this, take the module and start exercising from NEET beginners section. Okay. That's it. Thank you, ma'am.